guys, welcome back to Vital Signs. Today we're talking about cells. If you're new here, I'm Professor Thomas. I'm here to give you a crash course on all things anatomy and physiology. Let's dive in. Human cells are going to vary considerably in size and shape, but they're all going to be microscopic. They're all going to have cytoplasm and they're all going to have organelles. They'll also all be surrounded by a plasma membrane. So the plasma membrane is made of a phospholipid bilayer. We talked about phospholipids when we were in our chemistry section. Phospholipids have this polar head, a hydrophilic head, and two tails that are hydrophobic. Hydrophilic means it loves water, hydrophobic means it hates water. These two layers of phospholipids form an outer boundary to the cell. That's not the only thing we have going on in the plasma membrane. We also have a lot of proteins, cholesterol that contribute to the fluidity of the membrane and the stability as well. There are other proteins there that function in transport and signaling and self-identification. So there's a lot. The cytoplasm is a dense gelatinous matrix made of 70 to 80% water. So pretty watery, but not only water here, right? It is enclosed by the plasma membrane. So the cytoplasm is going to hold all of the organelles in the cell, things like the nucleus and the endoplasmic reticulum, the Golgi apparatus. These organelles aren't just floating around in this gelatinous matrix. They are held in place by the cytoskeleton. The cytoskeleton kind of gives the inside of the cell a framework to work with, almost like the scaffolding around a building. If you've ever been in a city and you see, oh, they're doing construction over here, you know that scaffolding goes up and down. The cytoskeleton functions similarly. It's not very set in stone. There's going to be moving pieces to it, but it does give some sort of structure or stability to the cell. The three components of the cytoskeleton I want you to know are actin filaments or microfilaments, intermediate filaments, and microtubules. The microfilaments or actin filaments polymerize in sort of a helical fashion. So those are kind of smaller, skinnier, weaker, if you will. Intermediate filaments are more stable than microfilaments, and they organize the tridimensional structure of a cell. Microtubules are comprised of tubulin, and they're hollow cylinders of varying length. Centrioles and centrosomes are also technically a part of the cytoskeleton, but we'll get into those later when we talk about cell division. Now let's start to talk about some organelles, starting off with ribosomes. Ribosomes are protein-making factories. Sometimes we'll find them attached to the rough endoplasmic reticulum or rough ER. Other times they're just free-floating in the cytoplasm. That rough endoplasmic reticulum, there's actually two of them. We've got a rough one and a smooth one. The rough ER, since it has ribosomes on it, kind of functions like a protein-making factory, where it's being used to collect, fold, and transport those proteins that the ribosomes on its surface are making. The smooth ER is smooth. It doesn't have those ribosomes. So it's going to be used to synthesize chemicals and make new membranes. The Golgi apparatus is a group of flattened sacs near the nucleus. It collects chemicals into vesicles that move from the smooth ER outward, and it also is a chemical processing and packaging center. The disc-like sacs that make up the Golgi apparatus are called the cisternae. So think of it this way. If the rough ER is the manufacturer of some kind of a product, then the Golgi apparatus is the Amazon warehouse of the cell, right? They're not really making anything at those Amazon warehouses. They're just getting them from manufacturers, packaging them, and shipping them to their final destination. So that's how the Golgi apparatus works. It isn't making anything per se. It's just taking those things that the endoplasmic reticulum made, packaging them, and sending them off. One thing that the Golgi apparatus does produce are lysosomes. Lysosomes are the garbage men of the cell. They are filled with digestive enzymes that are going to kind of absorb any sort of cellular debris that is present, break it down, and take care of it. Now let's talk about everybody's favorite organelle, the mitochondria. You probably heard the mitochondria is the powerhouse of the cell, probably in a high school science class. The reason we call it the powerhouse of the cell is because it makes ATP. You do need to know what that stands for, adenosine triphosphate. This is cellular energy. And to know that it is made in the mitochondria is not enough at this point. The mitochondria is made of two membranes. There's an outer one and an inner one. ATP is made in the inner mitochondrial membrane. Another cool thing about ATP is you kind of have to think of it as free money. If you walk down the street and there's money on the sidewalk, you're gonna pick it up and spend it, right? ATP is the same way. We can't just make the cellular energy and put it in the bank. Somebody's gonna pick that up and spend it. So as soon as ATP is made, it is being used. One last thing about the mitochondria is mitochondrial DNA is separate from the DNA of the rest of the cell. And all of the mitochondrial DNA that you have comes from your mother. The sperm's mitochondria gets destroyed as it's impacting against the egg during fertilization. So therefore, dad has no ability to contribute any mitochondria. So all mitochondria is maternally passed down, which is really cool. If a cell needs more energy, the mitochondria will divide via binary fission. A lot of textbook pictures of cells will only have one mitochondria in the picture, which makes you think, oh, there's only one mitochondria in the cell, but that's not true. If the cell needs more energy, the mitochondria divide, and now we have two powerhouses of the cell, 3, 4, so on and so forth. Going back up to the surface of the cell, we do need to talk about some surface appendages. We have microvilli, cilia, and flagella. Microvilli are these finger-like appendages that will be there to increase the surface area of the cell. So we find microvilli in a lot of intestinal cells where we're trying to absorb the nutrients from our food. Cilia are these hair-like appendages that can be there for sensory purposes or maybe to push something off of the surface of a cell. So we'll find cilia lining the entire respiratory tract because as we're breathing in and pollen and dust is getting caught in the mucus lining our respiratory tract, the cilia kind of push those irritants up until you can swallow them back down into your stomach. Flagella are a long tail-like appendage and they usually are there for movement. The tail of a sperm is the only example we have for flagella in humans. Now we'll talk about the nucleus. The nucleus is the brain of the cell. This is where we have our DNA, our genetic material. DNA is the instruction manual for everything that the cell is going to do. And DNA is stored there in the form of chromosomes. We have 46 of them. Structurally, the nucleus is covered by this nuclear envelope that has little holes in it called nuclear pores. And nuclear pores are how we get genetic information out of the nucleus to be read and used by the ribosomes as they're making proteins. Getting into membrane transport now, we have to get things into and out of the cell, and we do this in different ways. There's passive transport and active transport. Passive transport is going down a concentration gradient. So we're going from a crowded space to a less crowded space. And that is an easy process to make happen. We don't have to use any ATP to make that happen. Active transport is going against a concentration gradient, where we're taking something from a less crowded space and we're pushing it into an already crowded space. Nobody wants to do that. Nobody wants to go from a less crowded room into a more crowded room. And so how do we make somebody do that? We bribe them. We use the ATP to say, come on, get in the crowded space. That's active transport. Going back to passive transport, there are different types we want to learn. Diffusion is your bog standard passive transport, where we're going from a high concentration to a low concentration, no ATP necessary. Osmosis is passive transport, but specifically involving water. Dialysis is passive transport, but things are going across a selectively permeable membrane, meaning they don't allow everything to get through, only what they want to get through. And then lastly, filtration is a form of passive transport that happens due to pressure differences. Active transport is going against a concentration gradient. We need to know 3 different examples: phagocytosis, penocytosis, and ion transport pumps. With phagocytosis, the entire cell is engulfing something solid. That's active, it takes energy to make that happen. Penocytosis, the cell is engulfing something liquid. How I remember that is Pinot Grigio. It's a liquid, right? And I'm engulfing it. Then we have ion pump transport. The one pump that you want to be super familiar with is the sodium-potassium pump. Let's talk about it. Outside of the cell, we have a high concentration of sodium already. Inside of the cell, we have a high concentration of potassium, which is K+. So this sodium-potassium pump is an active transport pump. We're taking sodium from inside of the cell, we're pumping it outside of the cell where it's already super crowded. We're taking potassium from outside of the cell, we're pumping it inside of the cell where it's already crowded. So when we're pushing things into areas where the concentration is already high, we have to use ATP to make that happen. So how does this apply to your career in healthcare? Well, let's talk about a disease, cystic fibrosis. Cystic fibrosis is a mutation in the CFTR gene, which codes for chloride ion transporters. So when this gene is mutated, chloride ion transporters aren't working anymore. And when chloride ion transporters don't work in the cells, it results in a lot of mucus building up in the lungs and the pancreas, and that's going to manifest as a lot of respiratory and digestive system issues for these patients. Another example would be cholera. Cholera is a bacterial disease that messes with chloride ion transporters in the intestines. When we mess with our chloride ion transporters in the intestines, we get diarrhea, which is going to lead to severe water loss. Getting into cell growth now. The cell is only going to do one of two things: grow and be making proteins, or divide and make more cells. So the cell is growing by making proteins. How the cell knows what proteins to make is through the DNA that is in the nucleus. DNA is an instruction manual. It is a book of saying this is what protein you need to make. So if we have a code that's ATTCCG, we're going to read that information and know exactly what we're making. The reason cells can grow is because of the help of RNA. We have 3 different types of RNA. mRNA, which is messenger RNA, tRNA, which is transfer RNA, and rRNA, which is ribosomal RNA. Protein synthesis involves 2 steps: transcription and translation. Imagine this. You're in Egypt. You've discovered some hieroglyphics. It's 100 degrees. Are you going to sit down in the desert and start translating those hieroglyphics as you're in the heat? No, we're not going to do that. We're going to make a copy of the hieroglyphics, maybe take a picture, then go back to the air conditioning and translate them in there. That's kind of how transcription and translation works in the cell. We're not going to be making the proteins in the nucleus where the DNA is located. We're going to make a copy of the DNA, get it out of the nucleus, and then make the proteins out in the cell. So we've got a location change happening. In transcription, we're in the nucleus. We're taking that double-stranded DNA, unwinding it, and making a copy of that information. That copy is on mRNA. mRNA is exiting through a nuclear pore. It's going out into the cytoplasm. That's it for transcription. For translation, we're in the cytoplasm, and we are taking that mRNA, fishing it through a ribosome, the protein factories we talked about earlier. The ribosomes are made of rRNA. As mRNA goes through this, every 3 bases on mRNA is called a codon. So maybe we have AAT, that would be a codon, those 3. The codon goes through the ribosome and it gets matched with an anticodon on tRNA. The anticodon on tRNA matching with the codon on mRNA brings in an amino acid. Proteins are made of amino acids. So we're fishing this line of mRNA through. tRNAs are constantly coming in with new amino acids, and we're building this chain of amino acids that folds and gets more and more complex until finally we have a protein. That is how proteins are made. If the cell is not growing and making proteins, then it is dividing. We call cell division mitosis. The steps in cell division are super important. PMAT, that's how all of my students remember it. Prophase, metaphase, anaphase, telophase. PMAT is how we remember the order of those events. The goal of cell division is to get 2 identical daughter cells. When the cell is not dividing, it's in interphase. It's growing, it's making proteins, all of this stuff. Then it switches over to cell division, which is mitosis. In prophase, chromatin granules are becoming organized. Chromosomes appear and centrioles move away from the nucleus. The nuclear envelope is going to disappear and spindle fibers start to appear. In metaphase, chromosomes line across the middle of the cell, middle. Spindle fibers will attach themselves to each chromatid. In anaphase, centromeres break apart. The separated chromosomes are pulled to opposite ends of the cell. A cleavage furrow will start to develop. In telophase, cell division is basically complete. The nuclear envelope is reappearing, and the cytoplasm is dividing in cytokinesis. The end result is you have 2 identical cells. A couple of terms for you as we wrap things up. Hypertrophy is increasing the size of an individual cell. Hyperplasia is increasing the number of cells. Anaplasia is increasing the number of cells, but these cells are undifferentiated from one another, and they're kind of abnormal. Atrophy is the cells are decreasing in size. So if you don't use your cells, they tend to atrophy. For example, if you're in an accident and you stop using the muscles of your leg, you'll notice those muscles start to get smaller because the cells are atrophying. And there you have it, guys. That's the cell. That's everything you need to know. Thanks for joining me on Vital Signs. I'll see you next time.